opponents' workability issue, they stated that our policy won't work since it limits the progress of stem cell research. Our policy does not limit the progress of stem cell research. If our opponents meant to specify that our policy limits embryonic stem cell research, that would still be a false statement. Since, according to Cynthia Cohen, an article written by Cynthia Cohen, she stated that Bush did not seek to ban the pursuit of embryonic stem cell research with private funds. Bush stated that this allows us to explore the promise and potential of stem cell research without crossing a fundamental moral line by providing taxpayer funding that would sanction or encourage further destruction of embryos. In addition to that, Bush's policy forced scientists in this field to use their critical thinking skills to come up with alternatives to human embryonic stem cells without destroying human life, in which they did and are continuing to find. The domestic policy states that the dramatic advances in stem cell research since 2001 are evidence that the President's balanced policy is working. Our opponents also state that our policy has disadvantages. The disadvantages of using the alternatives to embryonic stem cells cause a complication to the host's health. Our opponents stated that a disadvantage to adult stem cells is that none can be used to develop treatments for humans, since all were originally grown on mouse cells, and they could expose humans to an animal virus that our immune systems could not fight. However, because they also state that adult stem cells are readily available, adult stem cells can continue to be researched to overcome that complication, which is a small complication compared to the ones I've stated before in embryonic stem cell research, where they have immune suppression and benign tumors that should convert into malignant tumors. Our opponents also mentioned how adult stem cells have limitations, such as not being versatile as embryonic stem cells. But the Domestic Policy Council states that given the right conditions, certain kinds of adult stem cells now seem to have the ability to differentiate into a number of different cell types. If this differentiation of adult stem cells can be controlled and sustained in the laboratory, these cells could become the basis of therapies for many serious common diseases and injuries. Our opponents also stated that another disadvantage of using adult stem cells was that the number of adult stem cells present in a person's body decreases with age. However, I have a source from the Humanities Model that states that the rapid death of transplanted embryonic stem cell-derived cells stands in striking contrast to the robust survival of adult cells when transplanted to adult tissues. Lastly, our opponents state that induced pluripotent stem cells have a tendency to transform into cancerous cells. But according to Louis Andrew, an article written by Louis Andrew, these induced pluripotent stem cells had a tendency to transform into cancerous cells, but this problem appears to have been short-lived. Because on November 30th, 2007, Shinya Yamanaka of Kyoto University, the leader of Japanese research groups, published results in Nature Biotechnology declaring that he was able to reproduce the stem cell creation process and eliminate the cancerous tendency by eliminating one of the 4 genes originally used. In the first process, 6 of 36 mice injected with the cells died of tumors within 100 days, but in the second process, no mice died. These alternatives seem more promising than embryonic stem cell research.